Well, hey friends. So this is a molecular genetics lecture that is kind of designed to provide some answers to a paper I'm going to give you and some questions I'm going to give you on that paper. And it may very well be our one of our last lectures. Hey, there you go. All right. So we were talking about prokaryotic transcriptional regulation and all that different kinds of models of positive control and negative control. And the pretty much basic tenet there is that we're going to take advantage of the fact that transcription and translation are coordinated. So the tryptophan biosynthetic pathway uses that. The other idea is that the RNA polymerase has an intrinsic binding affinity to DNA, and when it has a sigma factor attached to it, it has intrinsic binding affinity to promoters. So another control system is to have a not great promoter that requires an alternative sigma factor-like molecule. Another really good way of working this is to have some sort of physical block on the promoter or just downstream, and so the RNA polymerase can't proceed through. So that's a fairly fundamental, not very abstract way of thinking about gene regulation in the prokaryotic system. And in general, prokaryotes are probably drooling out small amounts of RNA and subsequently small amounts of every protein all the time. In the eukaryotic system, we have a much tighter regulation. First off, we have the separation of transcription in the nucleus and translation in the cytoplasm. So that means that there's a lot of steps between transcription and the actual production of the protein. Additionally, we have processing events like splicing, and we also have ways of taking RNA and storing it in a ribonucleoprotein and putting it in one end of a cell or another so that maybe it's not translated until some sort of signal. So there's a lot more control. One of the critical controls, though, that I'd like to talk a little bit about more is the idea of transcription factors. This is still a really critical role of gene regulation in developmental biology and in just general cell differentiation. And it goes kind of like this. We've got, for every gene, we've got the upstream regulatory region. There's going to be +1. This is, the same sort of conventions we use for our bacteria, in which we have the structural gene that is going to be transcribed. Of course, this then gets polyadenylated, and we can also have splicing occurring, and there's going to be the 5' cap added. So there's a lot of stuff that's going to be happening after transcription starts, but it is the initiation of transcription that is a fundamental critical point. So when we start looking at dissecting the upstream regulatory region, we can define two spots. We've got the promoter region, which is going to be a set of nucleotides that are fairly consistent. I mean, we have sort of a generic eukaryotic promoter. And that will allow the RNA polymerase to interact. We have, as we were talking about the different systems involved in eukaryotic transcription, the basal transcription apparatus, tau binding protein, and those various kinds of TFs bind here and establish a little nest for the RNA polymerase. But the real critical trick to having actual good high levels of gene expression is going to be a sequence that is typically upstream, but it doesn't have to be, known as, of course, the enhancer. And the enhancer was identified actually first in viruses that infect eukaryotes, and they've got very potent enhancers. And what was found is these are able to work in either direction, that is, their orientation and distance independent. So the promoter is a definition of something that is orientation specific because it directs which way the RNA polymerase is going to go, and it is distance specific. It is distance dependent because it again is establishing where +1 is going to be. And so if you move it around, you change where +1 is. An enhancer, on the other hand, isn't constrained by either one of these conditions. It can be pretty much on either side. It can be actually quite a distance away, upwards of tens of thousands of nucleotides. Hopefully, in a gene you're working on, it won't be that far away. And so understanding about the enhancer sequence becomes a really critical thing. And some of the very early stages of looking at eukaryotic enhancers and how they work was done in yeast and a variety of different kinds of microorganisms or single-cell organisms. And then people began looking at more advanced eukaryotic genome systems, like in mammals, and identifying genes that were associated, or regions that were associated with cell-specific transcription. And a classic one was the identification of a muscle cell transcription factor. And these are proteins that are found in muscle cells, and it turns out they are sufficient for causing a naive fibroblast to convert to a muscle cell. And there are two, the very first ones. One was called MyoD, and another one was called myogenin. And these are gene products, proteins, that bind upstream of muscle-specific genes and turn them on. And so very early on, some of the experiments were done in which you would take the MyoD gene or myogenin, and you could inject the RNA or even the protein into a cell, and that fibroblast would then convert into a muscle cell. So a couple of things that are happening here. That the transcription factor, we'll just call it the TF, it is binding to this spot on the gene sequence, on the DNA, on the chromosome. It is causing a folding of the DNA that allows for a much higher interaction of RNA polymerase with the promoter, and it allows for transcription, for efficient transcription. So a lot of work began looking at transcription factors. And one of the first things that was identified is that transcription factors can operate, or they have two specific domains. And it's an interesting way of thinking about proteins is this functional domain idea, is that the protein itself has regions that fold up, can carry out some kind of function, and there's another region that folds up and carries out its function. So the protein as a whole doesn't have to fold up into one large functional molecule. There's these little spots of function. And it turns out then, with transcription factors, most transcription factors, proteins, can be broken up into two different functional groups. We've got the DNA binding domain, and then we've got the transcriptional activation domain. Now, fairly early on in the analysis of transcription factors, these sort of domain swapping experiments were done to demonstrate that, yes, you can take a yeast transcription factor and isolate its transcriptional activation domain and fuse that with the DNA binding domain of another transcription factor. And you can essentially make a super activating transcription factor that binds to a very specific promoter sequence. So enhancers then really are nothing more than the DNA sequence that binds this DNA binding domain. So a couple of terms just to throw out. When people began looking at different kinds of transcription factors and looking for similarities, looking at homologies, a couple of things start to fall out. Is you can begin grouping transcription factors based on their DNA binding domains. They exhibit what we call a motif or a structural similarity. So a motif would be a sequence of amino acids, maybe not identical, but they're going to be very conserved, that typically form up into some kind of structure that allows for the binding of DNA. And there's a variety of terms that are probably worth throwing out here, just so when you see them in a paper, you'll know, oh, this is talking about a DNA binding motif. Not surprisingly, there are, just a set of amino acid sequences that have the ability to bind DNA. And they typically have a positive charged spot, because a stretch of positively charged amino acids. DNA being negatively charged, that makes sense. A classic transcription factor motif is one known as the basic helix loop helix domain, or known in the biz as a BHLH. This is a stretch of amino acids that forms an alpha helical stretch. There's a kind of a spacer and then another alpha helical stretch. They're able to fold around the DNA, and they have a set of very positively charged or basic amino acid residues. And so we have the BHLH, the basic helix loop helix. There's another similar motif that's called a helix turn helix. Then there are transcription factors that are named for different kinds of mutations that they produce. There's a transcription factor class that are known as the homeobox class, and they're named after a set of mutations that occur in flies. So this is a way we can begin to identify different kinds of protein sequences, amino acid sequences that can bind DNA. So there's a whole range. There's another motif that's known as the leucine zipper. So you got the HLH and the HTH, leucine zipper, homeodomains. There are a variety of different names that people will come up with. And that is a set of amino acids, an amino acid sequence, a domain, also known as a motif, that you can organize. You can begin to classify them based on their sequence homologies. Now, it turns out that the transcriptional activation domain, there's not a real good set of homologies. Primarily, transcriptional activation domains are mostly just an acidic blob. It doesn't have a whole lot of structure to it, and it doesn't have a lot of sequence homology across different transcription factors. It is primarily just a set of acidic amino acids. Now, this establishes the idea of the transcription factor, in that we've got the DNA binding domain and the activation domain. So now, in order to begin studying how these transcription factors, I'm looking at the PowerPoint stuff I'm going to send you to see if there's anything I need to throw in there, but so far we're doing good. There's a lot of remodeling stuff that is not all that important to our discussion in terms of histones and being able to open up the DNA, but primarily this is the main story. All right, so in order to study transcription factors, this becomes a really key sort of scientific experimental system if you want to understand how genes are being regulated in different tissues. That is, what makes a neuron, in that it's got a set of genes that are neuron specific. How are they being turned on? By transcription factors. It's not quite that simple, but it's a pretty good place to start anyway. That transcription factors are kind of making things go. Now, just to get esoteric, if you're taking developmental biology, cell signaling is controlling the activation of transcription factors, which then in turn is controlling the gene expression. But there you go. All right, so how do we experimentally go about identifying transcription factors? Good question. All right, this is an experimental system that is, with the age of genomics, it's not being done quite as much as it used to be, but it's still a really important thing to think about. What you can do is take the nuclei of a cell type, say muscle cells or neuron cells. You can isolate the nuclei, and you would expect, and it is true, that the transcription factors are going to be in the nucleus. So you can very gently lyse the cells and spin down the nuclei, and then very carefully crack open the nuclei using very slight amounts of detergent and different salt concentrations, and you can release the proteins that are present in the nucleus. Now, what you then do is take a gene of interest. Say we've got the big toe gene that you've identified is a gene that's expressed only in the big toe. You want to find out what are the regulatory regions and what are the systems involved in expressing this. Okay, so maybe you want to put big toes on different parts of your body. Fine. Now, what you would do is take the upstream region and you begin sort of breaking it apart. And this is where you begin doing some recombinant DNA work, where one of the things you could do is, rather than having the big toe gene, you could swap out this for something you can see, like green fluorescent protein, and put it back into the organism and see if you can get a green big toe. So you have a chunk of DNA, say, 2 kb of upstream region. You fuse that to green fluorescent protein and put that back into the animal, and bada bing, it actually generates a green big toe. So then within this 2 kb region is the binding site for a transcription factor. Now, how do you go about identifying that transcription factor binding site? Good question. What you begin doing is breaking this up into pieces, and you carry out something that is called an electrophoretic mobility shift assay. It is typically referred to as a gel shift. And this is how a gel shift assay would work. I'm going to go ahead and erase here and get a little bit more room. All right. So with a gel shift, you've got a DNA fragment that you think might be a spot for the binding of a transcription factor. You've got the nuclear extract that has all these different transcription factors in it. So here's how you would set the experiment up. It's typically run on an acrylamide gel, and there's a whole series of controls that you do. The first thing you start with is a small, usually 50 or so nucleotide piece of DNA, double-stranded DNA, that you can either synthesize or clone out, whatever, and you label it. And it doesn't have to be in-labeled, you just label the heck out of it. All right, so it's radioactive. Now, if you've got, which we will just refer to this as the DNA fragment. So we're going to run the DNA fragment, also known as the oligonucleotide or the oligo. We're going to run this on our gel. And so if we have just the oligo, we expect it to run down here and you get this big old blob. All right? So this is the electrophoretic mobility spot for the DNA oligo. All right. Now, in the next lane, let's add the nuclear extract plus the labeled oligo. And here's what's going to happen. We're going to see something down here, but we're also going to see a new band up here. This is the gel shifted band. This is the electrophoretic mobility shift. What is happening here is we've got the DNA fragment, and as a binding site for a transcription factor, it's bound the transcription factor. Now, this runs at a slower rate than just the DNA. So consequently, we've got a gel shift, a shift in the mobility. All right, so that works. Now, the controls are where things get fairly clever. One of the things you would do is keep the amount of oligo constant, and you begin ramping up the amount of nuclear extract. And so this is often demonstrated like that, where you got increasing amounts of nuclear extract. And what you hope to see is that this band gets stronger and this one gets weaker. You got a whole bunch of DNA here and not much down here. And so you're able to say, okay, we can begin pulling and titrating out this transcription factor. By adding more and more transcription factor, we can pull out the rest of the unbound DNA. Okay, so that's a good control. There are a number of other controls that I want to go into. So we're going to just keep the gel, but erase things a little bit. Another really good control is a competition. Now what we're going to do is have nuclear extract plus our labeled oligo, and then plus unlabeled oligo. Now, in this case, we're going to put a whole slug of unlabeled oligo and compete for a binding. And essentially, by adding way molar excess unlabeled, we should be able to bring the label back down here and have very little up here, because the bulk of this is no longer being labeled. That's a really good experiment to identify that, yes, you have specific binding. So you do a competition assay. Now, another brilliant control is to do the same competition assay, but now use an unlabeled mutant DNA sequence. So we've got the nuc extract plus the oligo. Now the mutant unlabeled. Now what's going to happen here is the mutated sequence, the different sequence, is unlabeled, but it can't compete, it can't bind, because it doesn't have binding affinity. And so now we should expect to see not much down here and recover our gigantic band. That is pretty much the absolute perfect experiment, where you're running a set of competition assays with cold DNA, unlabeled DNA, and you've got either the binding form or a mutated non-binding form. And that works. So, once you've identified the DNA binding site, another thing to do is begin to identify the transcription factor itself. And that actually can become, again, another really serious project as you begin wading through all the different transcription factors. One of the things people have done is build a little affinity column using this DNA sequence and try and pull out the nuclear extract. The difficulty with transcription factors, though, is they're going to be present at very small amounts because they're acting at the level of the genome, and you only got 2 copies of this gene within the genome. And so you don't need a whole lot of this protein present for it to be completely functional. So one way that you do this is a screen for all the different kinds of transcription factors that have similar motifs. And then you look to see where these different transcription factors are being expressed. That's kind of a blind way. Otherwise, you just have to go digging around in the kinds of genes that are being expressed in that specific cell type. But because this gene is going to be expressed at very low levels, it's a bit of a trick. So as far as a project goes, it's a noble project, but not one that one should undertake lightly. All right, that's where we're going to stop for now. I'm going to put together a little a paper that is got a real clear set of experiments like this, and give you some questions on that, and that's going to be kind of our final. And then I'm also going to probably do another discussion about some fly genetics, just because it's one of my favorite lectures to do. But for now, that's all I know. Good luck, and I will see you cats later. Ciao.